Got your seat saved here. Nice.
What exactly does it mean when it says it changes the shape of the myosin its head with the phosphorylation? Like,
Really just like this, the like part that connects it to the longer part of the heavy chain there. So like going from this 45-degree angle to that 90-degree angle. So it's just like kind of like, it's the cocking of the myosin.
the cocking and the changing of the shape is the same.
Same thing, yeah.
Okay.
Yeah, yeah. Yeah,
Then when it goes into cross bridge cycling, it starts with the action potential on the muscle cells. And the T2 goes, the function of those is to just push the action potential further down into the.
get it deeper into the cell. Yeah, so it can hit all the sarcoplasmic reticula like down below.
Be step two, is T2 goes pushing the action potential deeper into
Um,
the.
after the action potential.
Action potential
Yeah, yeah.
in the cell T2.
That's not cross bridge cycling though. That is excitation contraction coupling.
Oh, I'm getting you confused and then cross bridge cycling. Okay. Okay, thank you.
Yeah. What's that?
Can we like abbreviate like just action potential?
Yeah.
Okay.
So,
Just
yep.
for sake of time.
As long as it's like an abbreviation that we've used in class, then it's fine. If it's one I don't recognize, I don't know what it is, but yeah. Yeah, for sure.
Writing out the steps, we just said that calcium binds to troponin. I think that basically opens the binding site.
Yeah,
Do we mention DHPR and the ryanite? Did we mention that at all in the deck?
yeah, we should. Yeah, this was just kind of like more detail before I didn't write that out when I did the big writing thing, but this, you would
This is
want
a
to,
big part.
yeah, you would want to mention that, especially in the difference between cardiac muscle and skeletal muscle.
Right.
Yeah.
It's to have DHPR.
Yeah.
Where exactly would that go? Between.
So that would be when the action potential is going down the T tubules and then it activates the sarcoplasmic reticulum to release calcium. It'd be right in that step.
Okay,
It'd be just kind of more details about how calcium's released,
okay, so that, okay, that's the
part of excitation contraction coupling.
the DHPR is triggered by the
By what?
by the action potential.
Yep,
And that.
and that one triggers the ran.
It's-
Yeah, got it.
Got it.
Good job.
Yeah. So then what happened?
Yeah.
Because I think she was an accident.
Want me to say depolarization does? Okay.
Like I know this is the neuron, this is the sarcolemma, this space, where's the calcium five?
That's just extracellular space. Yeah.
Thank you.
Mhm. That your question too? Yeah, all right. All right, keep it coming. Keep it coming. What else we got? Need some more? More people?
So when we were going through cross bridge cycling.
Yeah.
We ended with restart at step one to do
Uh-huh.
this whole cycle again.
Yep,
Is step one talking about the neuromuscular junction activation?
No.
Or are you talking about when the myo?
just like those 4 or 5 steps there in the cross bridge cycle. Yeah,
Okay, perfect.
good question. Yeah.
Thank you.
Mhm.
Yeah. Yeah, for all some of his questions from the.
You guys ready? Go do a couple? Yeah. There we go. All right, let's keep it coming. Kind of from the back, there's like a hole in it. Yeah.
Well, because it's the end of the, well, I guess. Them doing, get it, the admin class, suppose, must show. So it's essentially started from.
Put a couple steps if you got it, Michael. Yeah. You guys ready? You guys are next. Put a couple steps up there. Get us moving. Come on.
Good time.
What's that? Enjoying the show? I know, it's kind of fun. Huh? Wow. Actually, did you spell those right too? Pretty good. I think he did. Wow, I don't think he has notes either. He spell, I don't know if I can spell dihydropyridine without notes, but yeah. Seriously.
That's pretty much the same thing. It's a lot different.
Good job on your spelling without notes there, Michael. That was amazing. I don't think I can
Well,
spell dihydropyridine
I
without
looked
notes.
at my
I tried
computer and
before.
then
Yeah, that's
sped
good.
right over
I
there.
mean, it's kind of like phonetic. the
I
way-
can,
but
I can
it's like-
spell it. I can't say it though. So don't
Yeah,
ask
it
me
is.
about that.
Yeah.
So when that happens.
So after the active binding.
See, Michael put the details in there for you guys, just like you're asking about. Yeah.
It helps me understand everything.
Totally. No, it's good. Yeah. Here. All right, come on, let's keep it going. Someone's got to tackle cross-bridge cycling. That's where we're at. Girls are still sitting here. You haven't moved yet. Counting on you. Counting on you to finish this out.
It's open now. ATP is the start. So probably just.
Seriously not. Yeah.
And we'll be best friends by the end of this program.
Oh, so. Join this program because I knew you were. This isn't
Gonna have a lot of best friends. Yeah, it's true. You guys will be. Yeah. It's very true.
It passes best.
That's the best. That's so bad.
You gotta read what. Yes.
Thank you.
Nice.
Open notes right now. That's all. Here,
Yeah.
here.
Guys got this. Bring it home.
Sir.
Yeah.
At some point, the ADP is removed during the
Yeah,
process. So the
it's-
myosin head is cocked and then the recycling is from the,
It's kind of sometime after before the new ATP comes, but after the phosphate is released,
okay, so
somewhere
it just
between
kind
there.
of floats off. Okay,
Yeah, I guess it's kind of, it's like, it's all like happening in like milliseconds,
okay.
you know?
Yeah,
So,
yeah,
like,
that's
pretty-
all kind of
pretty quick,
instantaneous.
yeah, it's
Okay,
pretty quick.
okay,
Yeah,
okay. Awesome.
yeah,
Thank you.
Yeah, for sure. Okay. Yeah, think we got it? All right, good job, you guys. That's fantastic. Okay. I think everybody was kind of following along as we went. But it looked complete to me. Did it seem like anything was missing that anybody else saw? Or not correct? I think it was great. So good work there. The couple of things that we'll mention as far as some of these differences, right? Key things that are different between how cardiac muscle contracts. We don't have the dihydropyridine receptor there. If we're looking at this one, right? Instead, we have those L-type calcium channels, which are voltage gated. Just like the dihydropyridine receptors are also voltage sensitive, right? But the L-type calcium channels open up with the change in voltage and the action potential. That lets extracellular calcium come into the cardiac myocyte, right? The same thing with the smooth muscle cell too, actually. So both cardiac and smooth muscle cell have those L-type calcium channels that change shape with the action potential, and extracellular calcium comes in to both of those cell types, okay? When it gets into the cardiac myocyte, that's going to activate the ryanodine receptors on the sarcoplasmic reticulum, which then those open up and let calcium flow out of the sarcoplasmic reticulum, right? In the cardiac myocyte, now we're just kind of back to where we were in the skeletal muscle cell, right? We go to troponin and all that happens. But in the smooth muscle cell, it's a little bit different, right? In the smooth muscle cell, that calcium that's released, and we can go to that slide. This one here, the calcium that's released doesn't go to troponin because there's no troponin in smooth muscle, but it goes to a calmodulin protein, which just simply means a protein that's modulated by calcium, right? And that one gets activated, which goes down and activates this myosin light chain kinase. That myosin light chain kinase actually phosphorylates myosin. It phosphorylates the myosin instead of so the activation of myosin happens before, right? And then that's what activates myosin ATPase to kind of continue the breakdown of ATP in smooth muscle. So that's a little bit different as far as the steps go there, because yeah. Okay, and so that's just going to continue the contraction there as long as this myosin ATPase stays activated. Right? Okay, so those are the big ones. We didn't really talk about a sarcoplasmic reticulum with smooth muscle cell. There is and that's because it's a little bit more complicated. Some smooth muscle cells have a pretty robust sarcoplasmic reticulum, so we can rely on some intracellular calcium. But other smooth muscle cells are totally dependent on extracellular calcium. So it's just kind of a mix there. Okay, so I think that covers everything we were talking about here. Any questions about that? Okay, go ahead. Yeah.
Recycling.
Yeah.
Once you have the conformational change from ATP that's detaching the myosin head,
Mhm.
where does like ADP and the inorganic phosphate go after that? Are they both released?
Point me in the right direction here. Which number are we talking about?
Yeah, I think, yeah, like between 5 and 6.
Between 5 and 6.
Steps of
Yeah, so this is all happening like within milliseconds, right? This whole thing. So it's like very, very quick. But what it seems to be, the prevailing theory is that phosphate comes off first, which is what causes that conformational change, which is what causes the power stroke. And then very shortly after, ADP also comes off. That's kind of the prevailing theory with how it works.
The release of the inorganic phosphate that
Causes
detach.
Actually causes the power stroke, not detach. It's attached to the actin at that point, still attached to the actin at this point. It's the arrival of the new ATP that causes the detachment for between there and there. Yeah, good question.
So
Yeah.
basically, six, once the ADP is gone, it's just waiting for the new ATP.
Yep, exactly. They're just stuck together. That's where, right, if no new ATP comes here, then you're in that rigor mortis stage, right? Yeah, yeah.
When the phosphate comes off.
Yep, that's the key kind of initiating thing there. Yeah. And I would love it if you guys could convince Dr. Jansma to get into the biochemistry of this, and with all the models she uses and stuff, it'd be really cool to actually see it on that level. That's beyond my chemistry knowledge, but it would be cool to have her do it. We've talked about it before. We've just never gotten around to doing it together. But yeah, so okay. Other questions? Okay, well, we're going to dive into metabolism here, and creation of ATP. And so all of this contraction stuff we're talking about in all these different types of cells, none of it would happen without ATP, right? That's the driver of all of these contractions, right? And so it's all about this hydrolysis reaction, right? Combining ATP with water, we get ADP and phosphate, and then this kind of free energy that we're using to usually change the shape of a protein, drive some reaction, something like that, right? And so the main things that use ATP in our skeletal muscle cells, number one, the thing we've been talking about, that myosin ATPase, right? So that's 70% of the ATP usage is there. But then interestingly enough, about another quarter to 30% is just moving calcium around, right? We talked about those Circa pumps that pump calcium back into the sarcoplasmic reticulum. That's a huge part of this. And then the sodium potassium pump and some others are kind of make up the rest of whatever we have left of ATP usage in skeletal muscle. Okay, so a little bit more about ATP. Again, this is kind of just this common currency of energy across all of biology, right? Almost all organisms use ATP to some extent to drive chemical processes, but we can't store a lot of it, okay? This amount here, 20 to 27 millimoles per kilogram of muscle, is only enough to last about 3 to 5 seconds of sprinting, right? So we burn it up super quickly. But our body is incredibly efficient at defending that storage, right? It's very, very rare that we could actually deplete a muscle cell of ATP, right? Even though we use it in 3 to 5 seconds, right? Your body's immediately replenishing it as it's being broken down, right? And so skeletal muscle can increase its ATP production by 100-fold pretty much instantaneously, all right? Like within a split second, it's increased its production by that much, okay? And so even with repeated sprints, if you guys have ever done a Wingate test before, has anybody ever done a Wingate test? Maybe a couple, yeah. So it's like a repeated, it's a single 30-second all-out sprint on a bike at a very high resistance. But there's studies where they have people do like 6 to 10 of those in a row with maybe a minute or rest between. And even with those studies, ATP is not depleted completely. Some people throw up after one of those, right? So doing 6 to 10 of them is crazy. So that's just showing the body's incredibly efficient at not letting us run out of ATP in our muscle cells, okay? And so we got to match the demand. Yeah, so the formation has to match the demand. Okay, and so our 3 ways to do that include number 1, the creatine phosphate system, number 2, the glycolytic system, and number 3, the oxidative system. Okay, so we'll talk about each of those in some more detail right now. Questions about any of this so far? Okay. All right, so the creatine phosphate system is the simplest one, right? So it's the use of creatine phosphate, which is an amino acid derivative. And this allows us to gain or reform ATP very, very rapidly. It's really just a one-step reaction. So we have this creatine, which has phosphate attached to it, okay? by use of this enzyme, creatine kinase, we can pull that phosphate off and then add it to ATP, or add it to ADP, right? Which is then going to form ATP, right? So simple, simple reaction, happens very, very fast. When we're thinking about how long can this energy system last if we're doing like an all-out sprint, it's not long. It's somewhere around 10 to 20 seconds. Okay, so oxygen is not used. The activities that really exemplify this would be a very short sprint, like a 10-second sprint, okay, or more like a single movement. All right, so that'd be like a jump or a punch or a swing or a throw, right? Those types of like single muscle actions are really the things that exemplify the use of this energy system. And yeah, most like weight training type activities, right? So repeated muscle contractions that last only a short period of time. Yeah. It does, yeah. So we've got really three primary types of muscle fibers in our body, right? We've got our slow twitch oxidative fibers, and we've got our fast twitch oxidative and our fast twitch glycolytic fibers. So the thing that dictates whether they're fast or slow twitch is the type of myosin ATPase they have. So there's different forms of myosin ATPase that break down ATP. They're faster or slower. That's kind of what gives us the fast twitch or the slow twitch. So in those fast twitch fibers, especially the ones that are called the fast glycolytic fibers, they would tend to rely on this energy system much more than the other ones, but also the glycolytic system as well, which is that mid-range system. Whereas our oxidative fibers, our type 1 fibers, can definitely still do the creatine phosphate system, and they use it especially while they're getting kind of ramped up to full production, but they would be less reliant on that and more reliant on the oxidative system. So yeah. It's pretty much, there's like a hierarchy because the oxidative system is the most efficient, right? We get so much more energy out of it and we're not wasting energy. And so the body's pretty much always trying to use that system if possible, right? But if it's in a cell that doesn't have many mitochondria, it doesn't have access to oxygen as well, it doesn't have the ability to produce ATP through the oxidative system as rapidly, then it's going to shift more to these other systems that are kind of the backup plan, but needed because they help us move faster, right? So yeah. Other questions on that? Okay, so a little bit more about the creatine phosphate system. It can provide about 3 times the stored ATP in about 10 seconds, okay? Which isn't necessarily a lot, but it's a lot for that moment, okay? In any activity that's lasting 1 second or less is pretty much just going to be 100% creatine phosphate system. But then activities lasting as short as greater than 1 second, but up to 6 seconds, we're already also adding in the glycolytic system. All right, so anything lasting more than a second, we're not going to just use the creatine phosphate system. We're adding in this secondary energy system, the glycolytic system. It takes about 90 seconds to recharge the creatine phosphate system. Okay, which is really big part of where our recommendations for rest between sets during resistance training comes from. Right, supposed to rest about 60 or about 90 seconds to 2 minutes, right, between sets with resistance training. That's really to give our creatine phosphate system a chance to recharge for the most part. Okay, some of you are probably familiar with creatine supplementation. It's one of the most well-proven ergogenic aids. It really just gives us more energy to lift and to exercise. It doesn't directly build muscle, but it allows us to put a greater stimulus on our muscle, right? So then we can adapt. We can have a greater adaptation if we put a greater stimulus on, right? And so the question with all these, one of the questions with all these energy systems will be, how does it get activated, right? How do we kick it on, right? And the other question will be, what makes it fatigue, what makes it stop, right? And so the stimulation here, and you're going to see some similarities with all of them, but it's stimulated by an increase in ADP and phosphate, which makes sense, right? In the presence of broken down ATP, that turns this thing on. It makes it work, right? Which is good, because as soon as we start breaking down that stored ATP that we have in our muscle, we need to replenish it immediately. And so that's why the stimulus for it is broken down ATP. Okay. And the way it fatigues is depletion of that phosphocreatine, right? So the creatine with the phosphate on there, we only have so much of that stored in our muscle. And so when we deplete that, this energy system doesn't keep working anymore. Yeah, go ahead, Ava. Yeah, recharge. So pretty much it gets depleted. We probably should have started with the depletion part, right? So it gets depleted. Now we can't use this energy system anymore if we're still in the middle of a lift or a sprint, right? But we have to stop for a certain amount of time. And then those cells replenish it by putting the phosphates back on the creatine. And so when we've got more of that phosphocreatine available, that's the recharging process. So good question. And so back to that Wingate test. If you've done a Wingate test before, you may have noticed that the first 15 to 20 seconds, you're pedaling super fast, right? It's a high resistance sprint on a bike. You're pedaling super, super fast. Your RPMs might be above 100, maybe 150 RPMs. But then right about at 15 or 20 seconds, you hit a wall, and all of a sudden your RPMs drop by about 50. And that's when your creatine phosphate system got depleted, right? And now you're just reliant on the glycolytic system and some aerobic as well. We'll talk about that in a sec, but the aerobic system does turn on within 20 seconds as well. But it's a very noticeable change when you hit that wall and the creatine phosphate system kind of gives out. So, yeah. Other questions or anything on that? Okay. Yeah, go ahead. There is some training possibility, some adaptations to like some of these enzymes. So the creatine kinase enzyme, there's another one called myokinase, which does show some ability to increase in its levels inside of our muscle. And then diet can make a difference. If you're not getting any meat or anything that has creatine in it, then that can make a difference. But as far as just like training causing more storage of creatine, phosphocreatine, not so much, but definitely supplementation and diet make a big difference. And then the enzymes do help a little bit probably, but not to a huge extent. Yeah. Yeah. Okay. Okay, so let's talk about our next two systems. And so our next two systems would be the anaerobic pathway and the aerobic pathway. But both of them, if we're talking about usage of carbohydrates, start with glycolysis, right? So both of them start with glycolysis. Glycolysis is a breakdown of glucose to pyruvate, okay? We'll kind of talk about after pyruvate, there's this fork in the road, right? We can go, if the pyruvate goes through this enzyme, LDH, which is lactate dehydrogenase, then it turns it into lactate. And that's the end of the anaerobic system. That's the end, right? If pyruvate's able to go through this enzyme, PDH, pyruvate dehydrogenase, then it turns it into acetyl-CoA. And this is continuing on our aerobic pathway or our oxidative system. Okay, we can see that these steps are happening just in the cytosol of the cell, right? Once we get down to this part, PDH is located in our mitochondria. All of this below will happen in the mitochondria. Okay, so glucose to pyruvate is glycolysis. We have that fork in the road. We'll talk a little bit more about that fork in the road here in a sec. But the other two big steps in the oxidative system, we have glycolysis first. This is actually a separate step too from pyruvate to acetyl-CoA, Krebs cycle, and then electron transport chain is kind of our final step. So we'll talk about each of those. Okay, so glycolysis, this is the glycolytic system because we're ending with lactic acid down here, okay? since we know it's the anaerobic pathway, we don't use oxygen here, right? Again, we're starting with glucose, or the reason it's called the glycolytic system is because most of the time in our muscle, when we use this pathway, this anaerobic pathway, we actually start with stored carbohydrate as in the form of glycogen, right? So we're starting with glycogen. There's a special enzyme there called FOS, which just abbreviated as FOS, but it's glycogen phosphorylase, that helps us break down glycogen into that glucose 1-phosphate, and then we can just kind of start the regular path of glycolysis there again, but we kind of skip that top step. And so one thing to note is when you skip that top step, you use one less ATP, right? So you can kind of see the minus ATPs over here. These are the ATPs used, and then the plus ATPs here are the ones that we form during that process. So so we got 4 on the plus side there. So we made 4 ATP. But if we started with glucose, then we used 2. If we started with glycogen, then we used only 1. So our net ATP production from the glycolytic system alone is either 2 or 3, depending on whether we started with glycogen or glucose, right? So start with glucose, 2, start with glycogen, 3. Okay. Couple things to note. Any of the enzymes that I mentioned that I have written on the slide there, not the ones that are, not all the, you don't have to memorize all these intermediates at all. I don't expect you to memorize that, but I do expect you to memorize the enzymes that I've kind of pointed out and talked about what goes into it and what comes out of it. Okay, so glycogen phosphorylase would be one of those. The rate limiting enzyme of glycolysis is phosphofructokinase, or PFK, right? That'd be another one that's important to know. Rate limiting enzyme would just be kind of like the weak link in the chain, right? Like if we're going to speed this process up, then we better make sure we speed that one up, right? That's going to slow us down the most. Okay, and then again, we've got this LDH and PDH, right? LDH is going to drive the last step in this glycolytic system to turn pyruvate or pyruvic acid into lactic acid. Okay, so this is our glycolytic system, or anaerobic system. It tends to, if we kind of do an all-out sprint, again, this system will last somewhere between 2 and 3 minutes. Okay, so the activities that really exemplify the use of this system would be kind of our middle distance activities. 400 meter to the 800 meter, something like that. We have a couple track people in here, I know. Yeah, so the middle distance events are the ones that really hurt. They really can make you nauseous. They can, yeah, because it's like you're sprinting for 2 minutes, right? 2.5 minutes, or 1.5 to 2.5, something like that. So, and you're producing a lot of hydrogen ions when you use this system, right? And it's not just from the lactate production, it's from other steps along the way that we get lots and lots of hydrogen ion production, which drives our pH down. Which is really what ends up stopping us from using this system in the end. But we'll get to that in just a sec. But questions on this before we go to the next slide? Yeah. PDH, that's the one that's going to turn pyruvate into acetyl-CoA, which we'll talk about that one more when we get to the aerobic pathway, or the oxidative system. Yeah, go ahead. You don't need to memorize the intermediates, all these phospho 1, 6-diphosphate, all those different things, but I do want you to kind of know the starting point, right? So glycogen versus glucose, what happens there? How do they differ, right? I want you to know this enzyme is key in kind of starting the glycolytic pathway, the breakdown of glycogen, right? The rate limiting enzyme of the whole pathway, I want you to know, PFK, and then definitely LDH at the end here. And then, yeah, those are the main things there. Okay, so then a couple more details about the glycolytic system. It can provide about 12 times the energy that we have at rest, that stored energy, before it fatigues, okay? The creatine phosphate system was only 3 times, right? So it's 4 times that, okay? Yeah, its usage, it's going to be stimulated by, so its usage is increased by an increased concentration of ADP again. Oops. Also a MP, which would be just one more phosphate group taken off of that ATP, right? Adenosine monophosphate instead of diphosphate. Okay, so that's all just like evidence of we've broken down a lot of energy and we need to replenish it, right? And the thing is, both of these activate that phos enzyme. All right, so question is like, how do we stimulate this glycolytic system to start doing its job? We stimulate it by breaking down ATP that's stored there, and then the byproducts of that breakdown of ATP or ADP and AMP. And what do those do specifically? Well, they activate that phos enzyme to get it to start breaking down our stored glycogen, and then those intermediates just start getting sent down the pathway, right? And the main intermediate that's gonna drive this that we see is glucose 6 phosphate. All right. And then glucose 6 phosphate is broken down by the key enzyme right here, PFK. Right? So that's the one intermediate I want you to know, just because it's kind of these key steps, right? So we activate phos by broken down ATP. Phos leads to the formation of glucose 6 phosphate, which is what stimulates our rate limiting enzyme, PFK, right? And that kind of drives this whole process from there. Yeah, Alexis? Oh, here it is. Okay. Okay, so this one is fatigued by inhibition. Okay, it's not fatigued by depleting glycogen. Depleting glycogen is a problem, but our body doesn't really let us do that because then we couldn't escape from that dog or that bear that is right there, and it could get us, right? Our body wants to leave that reserve there all the time, so we don't really deplete glycogen like fully in our muscle. But when our glycogen levels start getting low, we feel terrible, right? That's one of the key indicators of fatigue is when our glycogen drops. But the way that we fatigue it, the way that we kind of stop this system from working is a decrease in pH. So this can be caused by hydrogen ion production. It can be also caused by inorganic phosphate production. There's some other metabolites that are released from all the steps in the middle. That's just dropping our pH. And mainly what that does is that inhibits enzymes. And it causes pain. Right? We feel the pain, which makes it harder to push ourselves. But just kind of biochemically, all the enzymes that are driving all of these steps, they're all sensitive to pH, right? And so when that pH drops, then it slows everything down, right? It gets to the point where we just can't push the intermediates through anymore because those enzymes aren't functioning properly anymore. So it's great, right? It's a self-limiting system, right? This is a system that's a big waste of energy because we only get 2 ATP out of it. We could get 32 if we did it the oxidative way. So the body just kind of limits how much we can use it by this system, right? It's a negative feedback system. Okay. Questions on that? Yeah, go ahead. Yeah.
Glycolysis, or are we?
It's kind of the step after, yeah. And we'll talk about that when we get to the aerobic system here in just a sec. But yeah, kind of just consider it like the step after. Yeah, yeah. Okay, go ahead, Ava. Yeah, you're good. Yeah, so the simple answer that's not really true is that it's all from lactic acid. But that's what's kind of like the easiest way to look at it. But we definitely produce some hydrogen. Pretty much lactic acid is immediately dissociates and loses its hydrogen ion. So we don't really produce lactic acid because it just immediately lets go of its hydrogen ion. So that's one source of it. But also when we build up that free inorganic phosphate, because we've broken down a bunch of ATP, that also has the effect of decreasing our pH. There's also the buildup of calcium isn't necessarily pH related, but when we get too much calcium built up inside of our muscle cells, some of it starts binding to enzymes as well, which changes their shape. So it's pretty much just kind of all these different metabolites. And with a lot of these steps, none of this is happening like perfectly, right? So a lot of these involve combining a hydrogen ion to NAD and things like that. And so none of those really happen perfectly. So we end up kind of spilling over with some extra hydrogen ions throughout the solution. And so there's lots of sources of that hydrogen throughout. But yeah, it's all those. Yeah.
Down. Cells with the body compensate by taking glycogen from the liver.
It compensates by pulling blood glucose out and then turning it back into glycogen post-exercise typically. And so that's why like, if you've heard of like EPOC or afterburn is kind of like a gym term for it, you know, your EPOC or your excess post-exercise oxygen consumption would be a lot higher if you've depleted glycogen, because all of the glucose you have in your blood is going to replenishing that glycogen. And so in that time period, you're burning fat for fuel. So yeah. Other questions on that? Okay. So we did that. Yeah. So just a couple things about the theta pyruvate, because it's kind of this it's kind of connected to this idea of lactate, and there were lots of myths and kind of misunderstandings about it. The general answer you'll get in most biology textbooks, as far as the question of what dictates which way pyruvate goes, does it go through PDH or does it go through LDH? Still, the general answer is what? What causes it to go one way or the other? Presence of oxygen, right? Yeah. But that's definitely true if we put a cell in a petri dish and we give it oxygen. It's going to go through PDH. If we don't give it oxygen, it's going to go through LDH. Okay, and that's kind of how those conclusions were made. But this debate happened such a long time ago. It is pretty shocking that it's still this way in the textbook. But this whole Brooks and Davis debate is something you can kind of look up. This was happening really in the 90s, and George Brooks is still a UC Berkeley professor who studied this stuff. But pretty much he was making the case that there is some level of resting lactate production. Okay, so when you measure just lactate in the blood, there's always some there. Okay, meaning that, okay, we probably have enough oxygen in most of our cells to use the oxidative system at rest. Okay, the other thing is the evidence shows that we never fully deplete oxygen from muscle cells. Okay, so studies were done where they actually like look at intracellular oxygen levels during super intense exercise, and it never looks like we completely deplete oxygen from muscle cells. So we've kind of got those two lines of evidence along with a bunch more, and the conclusion was, well, there's always oxygen present, so it can't just be about the presence of oxygen depending on which way it goes. And so, yeah, so we'll talk about what it really is probably about. And I'll just put it here, two kind of things. So demand for ATP and a cell's oxidative capacity. So those are probably the two determining factors, and we'll talk about them more in a sec. But if a cell is using up a bunch of ATP so rapidly that the oxidative system can't keep up, because it's just not that fast, there's a lot of steps involved, then we probably have to shift to those other two slower systems, right? But part of what dictates whether a cell can keep up is its oxidative capacity, right? How much mitochondria that cell has, how many of those PDH enzymes are present, right? And so we'll get to that a little bit more in a second, but, okay, so the primary determinant of carbohydrate oxidation is going to be our pyruvate dehydrogenase enzyme, that PDH, right? So again, PDH is the one we see it right here, okay? PDH is the one that turns pyruvate into acetyl-CoA. So meaning like, how do we determine whether we're going to use carbohydrates through the oxidative system or through the glycolytic system? It's really about the presence and activation of this enzyme, this PDH enzyme, because that's the one that we're kind of waiting on for it to do its job. If we wait too long, then it's going to go through LDH instead, and we're going to use the anaerobic system, okay? So it is activated by a couple of things, ADP again, and calcium. So that's nice, right? Because we're dumping calcium into our skeletal muscle cells. So it makes sense that that would help us activate the enzymes we want to activate to drive this process, right? And so then it's inhibited by the things that it makes, right? It helps us to make NADH and acetyl-CoA. NADH comes here, right? It comes out of that process of turning pyruvate into acetyl-CoA, and then obviously acetyl-CoA is the product, right? So when we get a buildup of those things, like I don't need to keep doing this because we've already got enough of those products, right? Okay, but then the big question is when PDH can't keep up with the amount of pyruvate that's coming down from glycolysis, that's when it's going to spill over into that LDH enzyme. Okay, and the PLNU alums who had me for exercise physiology are going to recognize these next slides, but I think they explain it well. So if you kind of think of PDH as a funnel, the idea is with a funnel is it kind of has a certain rate that we can pour things into the top of it, right? If we pour that liquid into the funnel too fast, faster than it can exit out the bottom, then it starts to overflow, right? So if we think about PDH as that funnel, and the thing that we're funneling into it is pyruvate, right? From activating that phosphorylase enzyme and sending all that stuff down, right? If we think about us starting like a real light exercise stimulus, right? Like we're just go from standing to just kind of walking slowly, right? That breaks down some of that ATP that's stored in our muscle, leads to some phosphate and some ADP present. That's going to activate our phosphorylase enzyme up here, right? Which is going to lead to that glucose 6-phosphate, which is going to drive everything down here to start producing pyruvate, right? So we get a little bit of pyruvate coming in. This would be like us pouring slowly into that funnel, right? We're just kind of pouring slowly. It's not overflowing too much, kind of fills up, but it's still being released down the bottom, right? And so we kind of get a little bit of that. It's going mostly to acetyl-CoA, right? Mostly to the Krebs cycle. That's with light exercise, right? We would probably still get a little bit of spillover into LDH. We still would increase lactate production a little bit, probably, but none of that would show up in the blood. It would just all be handled in those muscle cells, okay? But instead, if we just started sprinting, right? We just start sprinting instead, that requires a lot more breakdown of ATP, right? So this vigorous exercise stimulus. So we have a lot of extra ADP and phosphate because we just broke down way more of that stored ATP. So we activate way more of that phosphorylase enzyme, right? And now we're going to send down a lot more pyruvate, right? So we're just dumping everything into the funnel now, right? So dumping the whole thing in as fast as we can. Most of that is going to spill over to LDH, right? And make lactic acid. A little bit still going to be going down the funnel. We're still using the oxidative system, but for the most part, most of this has to spill over. So again, the two things that dictate which way this goes, number one would be how many of these funnels do we have? Right? And how big are they? Right? That's about our mitochondrial capacity, right? The more mitochondria we have, the more of the PDH enzyme we have. And then secondly is how fast do we need that energy to be produced? Right? So demand for energy plus the cell's ability to produce energy through the oxidative system. Okay. Questions on that? Let me stop that from doing that. Okay. Questions on that at all? So yeah, for sure, if a muscle cell runs out of oxygen, it's going to use the anaerobic system. That's not wrong. It's just probably not the most likely case that's happening when we're exercising. It can happen when we exercise at altitude. That's been proven for sure. When we're in a hypoxic state, our muscle cells do run out of oxygen, and that kind of changes what limits exercise for us then versus when we're at sea level. But in normal conditions, we probably don't deplete our muscle cells of oxygen very often. Okay. All right. So we've kind of talked about the glycolysis part, entering up to the Krebs cycle now. The other thing to note is that we're just talking about the carbohydrate part, right? We're not diving too deep into the fat or protein usage, but the oxidative system can use both of those as well. And really the way it works with proteins is we break those proteins down into individual amino acids, and they're going to enter the Krebs cycle at different places. Some of them get turned right into acetyl-CoA. So with the 20 amino acids, they kind of enter these steps in different places each. And then with breaking down fats, we kind of break it down into two separate parts. Part of it becomes acetyl-CoA. Part of it ends up in glycolysis. But we won't go into too many details on that. So. Okay, so let's move on to the last two steps in the oxidative system, which would be the Krebs cycle first. Okay. So before we get to the Krebs cycle, though, this step that we were just talking about, pyruvate to acetyl-CoA, right? One of the things we produce there that's key is this NADH, right? If we go back and look at glycolysis, we'll see we produced a couple NADHs there as well. Those are going to be the key things that drive the electron transport chain. And now for the first time too, we're seeing the production of some CO2. Okay, so some CO2 comes from this step, the pyruvate to acetyl-CoA step, which is again kind of between glycolysis and the Krebs cycle. Okay, so we made acetyl-CoA. One thing to note if we go back to the glycolysis or the glycolytic system one here is after this step, you see two arrows form, right? So we're breaking these into two intermediates after step, we're really at, yeah, after step 4 there, instead of having one intermediate, we have two now, which then results in two pyruvates down here, okay? So when we go to the Krebs cycle, we don't just have one acetyl-CoA, we have two acetyl-CoAs, and that's gonna make it so the Krebs cycle happens twice, right? Those two turns of the Krebs cycle, because we have those two acetyl-CoAs. Okay, so the most important things that come out of the Krebs cycle would be number 1, NADH and FADH2. Okay, so we see some NADH there, more there. We see some FADH2 there, and then some more NADH there. Okay, and then we also produce some ATP. Just right here, you can see some ATP production. And then also the byproduct of CO2. I think I meant to put these on two separate lines. That's probably why I had 3 bullet points, NADH and FADH2, but yeah, and then this is our other source of CO2, right? So some CO2 here, some CO2 here. This is the CO2 that we breathe out. One thing to note, the rate limiting enzyme here is called isocitrate dehydrogenase. We can abbreviate that as IDH. That's the rate limiting enzyme in the Krebs cycle. Okay. And kind of the derivatives or the things that kind of make up the backbone of these NADH and FADH2 are vitamins that we consume. Anybody know which vitamins they are? Anybody got a nutrition background? No. So NAD, I want to make sure I don't mix them up. NAD is always, that's B3 or niacin. And FAD is B2 or riboflavin. So key vitamins that are needed for energy production because they're kind of part of the makeup of these, of this NADH and FADH2 complex. Okay, go ahead, Alexis. Yeah.
Fit it out. You want to down.
Yeah, pretty much. We'll, in a sec, we'll kind of go through and count NADHs and FADHs and how those translate to how many ATP we get. So I want you to, we didn't necessarily do that here yet, but we will in a sec. But besides that, everything else that I kind of pointed out on the sides or talked about would be the key things. So yeah, you don't need to memorize every step in the Krebs cycle. Yeah. Yeah. How many people had to memorize every step in the Krebs cycle for an undergrad class? Yeah, so you guys are well suited for all this for sure. I like to try to point out the key things and maybe understand it more than just memorize everything, but that's kind of what I'm going for. But other questions on this? Okay, all right, so then key here is we made that NADH and the FADH2, and those things, those molecules are going to make their way to this inner mitochondrial membrane. Okay, so when we think about our mitochondria, it has phospholipid bilayer like this, but then it has another phospholipid bilayer up here. Right, so there's kind of these two phospholipid bilayers, and so there's this space in here called the intramembrane space, right? The space between those two membranes. Okay, can you kind of see it here? Yeah, kind of in this one. All right, this is supposed to be a mitochondria. So we kind of got one membrane here, right? And then another membrane here. So where all those hydrogen ions are is kind of in this space between the two membranes there. Okay, so now we're to our electron transport chain where all the fun happens. Okay, we got NADH delivering electrons. We got FADH2 also delivering electrons. Okay, and the whole deal here is that it's very negatively charged up here and much more positively charged down here. Okay, and so as we drop these electrons off, we have these different carriers, protein carriers that allow the electrons to flow down this direction. But as they're flowing down that direction, there's energy in that electrical gradient pulling those electrons down the membrane. Okay, and so the energy for that electrical gradient can be used to take hydrogen ions and pump them into that intramembranous space. Okay, there's already a lot of hydrogen ions up here, right? There's already a lot. There's not many down here. So we can use the energy of this electron gradient to pump hydrogens up this way. What form of transport would that be from the ones we talked about last week? Which one? Secondary active transport. That's it, right? So this is secondary active transport. Wouldn't make any sense if we use primary active transport to do this whole thing, because the whole point is to make ATP, right? Not use it. So we got secondary active transport at play here, right? So we're building up this big concentration of lots of hydrogen ions over here. They can't pass back through over here, even though they're trying to because of the concentration gradient, okay? Until they get to the end here, and then they rush through the key enzyme that drives this all ATP production for us for the most part, 99% of it at least, which is ATP synthase. So those hydrogens rush through ATP synthase, and there's a lot of energy behind that gradient. Okay, we have an electrical gradient, and we have a concentration gradient for hydrogen ions there. And if you extrapolate out kind of the charge difference across one side of the membrane to the other, those membranes are only about 1/1200th the thickness of a piece of paper. So it's really thin, right? But if we kind of took that really thin layer and extrapolated that out to like life size, something that we could picture and see, the charge difference is the equivalent of about 30,000 volts, like about the strength of a bolt of lightning. And so this is an incredible amount of power that our mitochondria can create and harness. And so it's using that power to push these hydrogen ions through. We'll come back to this slide in one sec. But when it pushes those hydrogen ions through, it turns this big ATP synthase, right? This is ATP synthase. Super cool enzyme for sure. Dr. Jansma could geek out on that for a long time with you guys for sure. But when hydrogen rushes through that thing, it turns it. And it's somewhat similar to a rotary engine, right? So if you look at certain vehicles like Mazda is kind of known for using the rotary engine in their vehicles. It's totally different than our typical like cylinders and stuff that we have in most engines. But it uses a lot of the same components that this little molecular machine does. And so hydrogen rushes through, turns this thing. And as it turns it, that's creating, it's taking chemical energy of that hydrogen ion gradient, converting it into mechanical energy, right? Which is just movement of a protein, right? And then that movement of the protein pushes the phosphate onto ADP, right? And then, so that's kind of converting it back to chemical energy, right? So we're going from chemical energy, mechanical energy, the movement of the protein, back to chemical energy in the formation of ATP. Okay, and so all of that happened because we created this big gradient of hydrogen ions up here, right? And then we let them rush through and kind of harness the energy in that. But the only reason we could do that is because we first had an electron gradient, right? And it's always so fun talking about all that stuff. Sometimes I forget to talk about oxygen, right? Which is kind of crucial here, right? Without oxygen, this whole thing stops, right? Because the key here is that oxygen is the final electron acceptor, right? And so this equation is key. The thing it's not really showing super clearly, but it is because it's showing it come here, is that we also have to add, let's do it this way. I'll just write out the whole equation. Let's go 2 electrons plus those 2 hydrogen ions plus half of an O2. That gives us water at the end, right? So that's the oxygen accepting those electrons. Because if we didn't have the electrons here, right? If this was just 2 H pluses plus half an O2, that would be H2O plus 2, right? Which isn't water, right? So we add those 2 electrons, so it's H2O, and that's the final step there, right? So as soon as if we don't have oxygen in our cells, this gradient that we had for electrons to move down goes away because the electrons start building up down here, right? And so now instead of this being nice and positively charged, it's negatively charged. And so we don't have this gradient anymore. So then we can't use that secondary active transport to push the hydrogen ions anymore. We don't have this hydrogen ion gradient. We can't make them rush through ATP synthase, and we can't make ATP, right? So all those things are kind of interdependent on each other. Okay, a couple key things here is we make 28 ATP here. Okay, we make some water. It's called metabolic water production. We need the NADH and FADH2 to drive this. And obviously we need oxygen. Okay, yeah. And one other little ratio that's important to know is that for every NAD, we make about 2.5 ATP. And for every FADH2, we make about 1.5 ATP. And so these two numbers, these little ratios, the 2.5 and the 1.5, this is where you get some variation in ATP production. Some of you in your college biology class, for sure, in your high school biology class might have seen that we make like 38 or 39 ATP for glucose. For fat, it was up a number as high as like 144. Now it's more like 106. It's because of these ratios aren't always static. They change based on the energy status of the cell and the temperature of the cell and things like that. And so it looks like it's less efficient than on average, than we thought in human cells. That's why those numbers have gone down just a little bit. But, okay, questions on that? Okay, so here's how we're going to do a little recap of that for the last 15 minutes of class. I want you to go through kind of starting back with the glycolysis, okay? This glucose to pyruvate, just kind of fill in all of these things. How much ATP was used, how much was made, NADH made, and then you're going to kind of add all that stuff up, right? How do we get to that number, that final number in the end of number of ATP? And yeah, I'll be walking around helping you guys. So go ahead and we'll spend the last 10 minutes doing that. I'll wrap it up in the last 5 minutes and that'll be it. So go ahead.
This should happen on the side as well.
It is inside, so, yep, good question. Yeah,
This. We have, if you start with glucose, you lose two ATP.
yeah.
If you use glycogen, you use 1 ATP.
Yeah, it's just because this step, going from glucose to glucose six phosphate, requires an ATP to drive that reaction. Whereas going from glycogen to glucose one phosphate to glucose six phosphate is energy free. Yeah,
And then is there like two glucoses? Oh, wait, no, it's just one.
just one. Yeah. But then the kind of tricky part is that it costs ATP to make glycogen, so it's like it
Yeah.
kind of balances out in the end. But
This, glucose uses 2 ATP, glycogen uses 1.
that's right. Yeah. No, that's right. That's the. So there's one here for glucose that doesn't have with glycogen, but then another one here for both of them.
Yes.
Yeah.
Okay, but then glycogen, it skips this part.
Skips that first one. Yep. Got it.
Reading it.
Yeah, totally. For sure.
Bit confused about
Yeah.
is, and like, we were talking about it. So for here, the channels, so is it going against its gradient or with its gradient?
Which one, the hydrogen ion?
The hydrogen ion.
It's going against. Yeah.
Okay, and that's going against because it's negative to positive here, or that's where. I'm
There's
getting-
some electrical gradient, but it's also concentration gradient too.
Okay, a little bit of concentration.
Yeah. Mhm.
Okay,
Yep.
so it's going against, and then would this be going with its gradient through the? ET
Yes, that
synthase?
goes with it.
Okay.
So it's kind of sealed up to the passive movement of hydrogen until we get to that ATP synthase, and then that's what lets it pass through.
Right, so should I imagine this as like both outer or like these areas, or how? do I imagine that?
No, so if we go, yeah, mind if I write it,
I don't-
draw it right there? Yeah, yeah, so if we have like a mitochondria like this. This isn't really what it looks like, but kind of, so the hydrogens are all,
Oh, it's because you hit the,
I hit the thing.
yeah, don't hit the,
Oh, this.
yeah,
ah,
if you hit the button.
Oh, I see. Yeah, I always mess up my Apple Pencil Pro as well, like that. So yeah, all the hydrogens are in here. Like this space is in there, right?
Mhm.
So that's like that intermembrane space. And then here is like, this is right there, right? This is this part right there.
Okay,
Is that up? And so all these enzymes are embedded in this membrane.
got it.
Yeah.
Okay. So all of the, and this is like passing between to this inner membrane.
Yep.
Okay, that
Mhm.
makes more sense.
Yeah.
Alright, thank you.
Yeah. Sure. Well,
Oh, sorry.
you're good.
See there. For glycolysis, I think I'm confused about the NADH.
Yeah, so if you go back to the graph, you can see we made some,
Here, is
okay,
it only the one?
there's one on each side here, so it is two. Yeah.
Two.
Mhm.
Okay, two NADH.
Yep.
Okay, and does that change depending on if it's glycogen or glucose?
Mm-mm.
Okay,
No,
okay, thank you.
yeah, sure. Yeah. Yep.
For that, I just didn't get all the details there, but.
For the NADH made for glycolysis, which has like the best picture of that, if any.
The one that says glycolytic system, which is this one. So yeah, so you can see NADH. there.
Okay, so that's where
Two.
it's made and two.
Yeah.
Okay, thank you.
That's 88. You got two right there. And that's two.
Three.
2.5.
Yeah.
The difference between the 2.5 and 1.5.
Why is there a difference? It's because of where it enters.
Uh-huh.
NADH enters earlier, so there's a chance to pump more hydrogens. Whereas FADH enters a little bit later, so we just don't have as many hydrogens pumped across for that. And then I don't remember the number, but it's something like 6 hydrogen ions needed for each turn of the ATP synthase or something like that. So that's kind of where the ratio comes from.
Okay. Initially, I would think there's more hydrogens that FADH could give, so maybe
Yeah,
there'd be
right.
more ATP.
I know.
It's
I
just
know what
based
you
on.
mean. Yeah, it's
the
based
gradient
on kind of where
goes.
it starts, yeah.
Okay,
Mhm.
got it. Thank you.
Yep.
Okay. Go to the. To which one? So it's. Electron transport chain? What do we need?
Something broke.
Nothing.
Oh, no.
Like, I can't shut it now.
Oh, shit. Oh, it's like something stuck. Maybe you just fixed it. Yeah, there you go. Good job.
Wait.
Good job.
Just fix it.
Nice work. You did it.
First.
Oh, yeah, there it is again. Yeah, it's like it needs to tuck under a little bit more or something. Yeah. Yeah.
Are you assuming this is we're just doing one molecule pyruvate in this one? This is glycolysis creates two molecules.
It creates two.
Yeah.
Yeah. So that doesn't change what you get in glycolysis, but it'll change the fact that you do 2 turns of the Krebs cycle
Okay.
because of 2 pyruvates and 2 acetyl-CoAs. Yeah.
Thank you.
Wait, can I see it? Yeah.
Doing good?
Yeah.
Yeah.
We use ATP and the system we're getting.
Yeah.
So is that just 6?
That's where I got it.
What do we got?
How we doing?
I think I got it.
Got it? Good.
Then do some multiplication.
Yeah. Math will get you in the end.
Yeah.
Yeah.
So the Krebs cycle does 2 turns, exactly. Because there's 2 glucose.
Nope,
No.
the 1 glucose breaks down into 2 acetyl-CoAs in the end, but also 2 pyruvates.
So the glucose splits into 2.
Yes, if you go to that glycolysis slide, you can see where that happens. So once we go from here, there's 1 fructose 1,6-diphosphate, and then all of a sudden there's 2 of these, yeah. So the rest of the way there's 2. Yeah, that's a good question. Yeah, yeah, it probably just depends on how much like enzymatic power the cell has, because it's just a bunch of enzymes going on. So it's probably it all going on at once, you know. So yeah.
So glucose splits into 2 pyruvates. Each pyruvate turns into acetyl.
Say that one more time.
Glucose turns into 2 pyruvates,
Yep.
and each pyruvate turns into acetyl.
Yep, got it.
They don't. They just.
This is split again, yeah. Mhm.
Thank you.
Yeah, for sure. All right, guys, let's come back together and just kind of wrap this up here. Make sure we're all on the same page. So we'll start up here with glycolysis, all right, which is glucose to pyruvate. Okay, the number of ATP used, what are we going to put in there? If we're doing glucose to pyruvate, 2, okay? If we're going glycogen, then it's 1, right? ATP made is how many? 4. NADH made, 2, okay? So then we got that next step, pyruvate to acetyl-CoA. We're going to make 2 NADHs here. Okay, and then we get down to the Krebs cycle, we end up with 2 acetyl-CoAs because, again, just to point this out, we're going from 1 intermediate here, and that one splits into 2 of the same intermediate, right? So then we got 2 of everything from there on out, so we got 2 pyruvates, which would result in 2 lactic acids, but also results in 2 acetyl-CoAs. Okay, so yeah, so we got Krebs cycle each turn. How many NADHs each turn? 3, okay, oops. How many FADHs? 3, 1. And then number of ATP we make, just 1 each turn, right? So we got 2 of these, so we're kind of just timesing all these by 2. Okay, those little ratios I gave you, NADH gives us 2.5 ATP per NADH. FADH2 gives us 1.5. The reason for that is just because of where they enter the electron transport chain. NADH enters earlier, so it has more complexes to go through, which allows for more hydrogen ions to get pumped in the end. And then it's kind of like the number of hydrogen ions converts to the amount of ATP you make, where FADH2 enters the chain a little bit later. So there's less hydrogen ions that get pumped across and a little bit less ATP. Okay, so then how many NADH total? How many do we have total? How many? 10, right? And the 10, yeah. So we got 10 NADH total because we had 2 here, that's 4. And then we had 3 here, but we times that by 2, so that was 6 actually, right? So 6 NADH from the Krebs cycle. So 10 total. So it's just 2.5 times 10, which would be 25. So that's where the vast majority of our ATP comes from, right? How many FADH2? There were 2 of them. Yeah, 2 of those, times that by our 1.5. So we get 3 there, all right? So there's our 28 between those 2 that we get from the electron transport chain. All right, ATP from the Krebs cycle, we had 2. ATP from glycolysis would be 2 if glucose. Well, this would be 4, this net number, right? But total here would be 3 here. So total from glucose would be 32. Total from glycogen would be 33. Okay, I think we got it. Yeah, go ahead. Yeah, because we start with 2 acetyl-CoAs and that's what starts it. So go through 1 acetyl-CoA goes through, you get all this stuff. The next one goes through, you get all that same stuff again. Yeah. Okay, other questions or clarifications on that? Yeah, Ava? Yeah. Yeah, and I wasn't mentioning the CO2s here, but we also get CO2 here and we get CO2 in the Krebs cycle. So those will be the 2 primary sources of CO2 that comes from metabolism, will be from the Krebs cycle and from that step of pyruvate to acetyl-CoA. So we get 6 moles of CO2 per mole of glucose, but I wasn't going to make you memorize that number. Okay, what else? Other questions? All right, yeah, got another one, Hannah? Yeah.
Glycolysis, what do you mean by like net, or is there like another terminology that you'd be using? I don't think so.
Yeah, net meaning that we make 4, but we used 2, right? So the net would be 2 there, right? So for glucose.
Oh,
And then for glycogen, we used 1, but still made 4. So the net with glycogen would be 3.
so glycolysis, like the net.
Yeah. Oh, sorry. Yeah, I guess shouldn't have put a 4 there, huh? Let's just go with these numbers. Here's our net there, and there's our net there. Yeah, thank you. Yeah, thanks for pointing that out. Yeah. Yeah, go ahead. Yeah, so we always have 28 from the electron transport chain, right? We always have 2 from the Krebs cycle, so that gets us to 30, right? And then if we started with glucose, our net ATP production in glycolysis is 2. So that gets us up to the 32. If we started with glycogen, our net ATP production in glycolysis is 3. So that goes to the 33. Yeah. Yep. Other questions? All right. 10:59. Have a great rest of your day, guys. I'll be hanging around a bit if you got questions. Okay, you're welcome.